Hi, everyone. I work here in the Bioscience Center. I'm Kim Finley, and I'm starting out with a really beautiful slide because the talk is going to become quite grim very quickly. I'm going to be talking about neural degeneration. So this is a huge problem, and it's only getting worse. I don't know if any of you watched the 60 Minutes program that was aired last Sunday, but they were really looking at a family that has early onset familial Alzheimer's disease, and it was really clearly stated in the program that there's not much out there. So it is a growing epidemic. There's not much out there in terms of treatments, and there's very little understanding as to actually how the disease develops and what we can do to potentially prevent that. So just to give you the grim statistics, there's over 1 million Americans right now suffering from Alzheimer's disease. And the cost in terms of economics is becoming quite staggering, and it's only going to get worse. This is including healthcare costs and also the amount of time and effort spent by families trying to care for their loved ones. So it's really quite something that's problematic. And there is some genetics understood about this, not a lot. And there's one of the risk factors for developing Alzheimer's disease is simple age. So I think the estimates are that if you are lucky enough to make it to 85 years of age, you're going to have about a 40% chance of having Alzheimer's disease by that point. This has been a really tough nut to crack. I mean, the best you can do in terms of understanding the disease in humans is to do post-mortem studies. So they actually wait till someone has passed away, and then they can go ahead and look at the development of structures in the brain called plaques and tangles, and it's illustrated up here. And this is a lot of, in terms of protein turnover is altered. And clearance of these things, it seemed to be really problematic for nerve cells. They don't like it at all. It really is problematic. There's other forms of neural degeneration. Again, a lot of those are age associated. You have Parkinson's disease. I'm sure most of you have heard of this. There's ALS. And more recently, there's a type of it's not quite like Alzheimer's disease, but it's the chronic traumatic encephalopathy that sports people have been really kind of coming to the forefront with that, and that's more of a tauopathy. So CTE is really kind of bringing to the forefront what's going on. Again, there's a lot in terms of genetics that's starting to be understood, but more recently there's an appreciation that an inability to clear protein aggregates from nerve cells is a real problem. And so, and again, the head trauma is also a risk factor for developing Alzheimer's, Parkinson's. You can think of Muhammad Ali on those kinds of things. So what we really need right now is a fast, efficient, cheap genetic model to move this forward in a reasonable way. And you just have to understand the genetics, and I'll touch in on some environmental things that we've been working on in the lab. The best thing you can work on is designing a system where you can find bad things as well as good things. So it's really nice if you can push that envelope that way. And this is a picture of an adult fly brain. This was taken here at the biology department. Thank you for the confocal microscope. This brain is stained for nerve cells. So this is highlighting that they're very complex. So I can pass around these little guys if anybody wants to see the fruit flies. They're here. You're welcome to look at them later on. And the genetics has been amazing. Basically, I want to really highlight what has been learned from fruit fly genetics is not just development. There's been, in terms of the nervous system, there's been all kinds of things in terms of how we see, how we touch, how we have olfaction, how we smell things, all kinds of complex behavior circuitry. All of the circadian genes were first identified in fruit flies, as well as the genes that are involved in learning and memory. So these are all fundamental things in fruit flies. I'd like to highlight that they have very fast development, so that takes about 11 days from an egg to a mature adult. And the genetics of fruit flies have been studied for about 120 years. So it's been around for a long time. They were the second genome to get sequenced. And right now there's probably well over 100,000 different fly strains that you can just go online and order, and they cost about $10 to $15 a pop. And in terms of aging studies, their average lifespan, so that's a mean lifespan, is about 1 to 2 months. It depends on what we do to the fruit flies. And just to highlight the history behind this, these three wonderful people won the Nobel Prize for Drosophila genetics in terms of understanding what is controlling early development. And this lovely gentleman down there on the right actually won the Nobel Prize for immunology, using Drosophila to understand the molecular mechanisms behind inflammation and the immune system. So, just so you didn't think it was important, we really do have a lot of similarities with fruit flies. So I came into this field of autophagy. So this is the main bulk clearance pathway that's going on in all of your cells right this minute. It basically involves making a bag around garbage, sealing it up, and transporting the garbage to the big recycling organelle in the cell, which is the lysosome. The contents of the autophagosome is put into the lysosome, and those component parts are broken down and actually recycled to the cell. It was first identified at the molecular level by this wonderful gentleman who just won the Nobel Prize for autophagy this year. And he basically used a yeast as his genetic model system. And whenever you are fasting, you activate this pathway. It's a cell survival pathway. Okay? In the nerve cells, what my research has shown is that it is a critical pathway for engulfing protein aggregates in nerve cells and eliminating them. If this pathway is not working, we start seeing really bad things happening. And that came from my cloning of a gene, which I got to name blue cheese, because they had little aggregates in the brain. And you can see that they actually, if you compare a normal fly brain with a mutant fly brain, they have atrophy fairly quickly. We're talking 11 days. And it's very similar to what you see in people with Alzheimer's disease, where you actually have atrophy. And then the other thing is that they start to have protein inclusions starting to form throughout the CNS. And again, these are the same markers that we use to look at that in human beings. So the parallels are amazing. Fortunately, blue cheese has a human homologue, which was named Alfie by some friends of mine in Norway. And we're now pretty clear that it's involved in autism and Huntington's disease. This is just showing that we can use this stain to highlight other autophagy mutants. And then we've gone ahead and we can actually do a lot of physical things to the fruit flies. We can look at lifespan, and we can see that the two mutant flies compared to the black line are not living as long. One is blue cheese, one is another mutation in the pathway. And we think it's pretty clear, though, this is using the same marker that we saw with the pictures. This is actually running those proteins out on a gel and staining for those. You can see this incredible buildup of protein aggregates. We can quantify this. We're always looking for quantitative assessment. Now, the cool thing that you can do with fruit flies is put things back. So you can not only just look at mutants, we can look and see if anything does good things for the fruit flies. And you can see the nice red line out there. We can actually just putting one gene back in the nerve cells of these fruit flies has them living 60% longer on average. And we are pretty clear that it's because they have lower protein aggregate levels. So based on this kind of analysis, we can look at for not only negative things that impact lifespan and neural maintenance, we also are up to finding positive markers or positive modifiers, and always wherever we can improve neural autophagy, we increase lifespan, we preserve behaviors, and we're looking at genetic factors that do this right now. We have a diet that is really doing some rather amazing things, and we've also identified some neuroprotective drugs. So this is taking our system one step further, and this is work by Eric Ratliff and I.A. Barquette, and this is our trauma model. So can we look at environmental causes of neural degeneration? And this is actually traumatizing the fly in a tissue homogenizer. This was done by the engineers at the company that built this. And we can actually expose the flies to exquisitely defined levels of trauma. So we have a high impact, we have mild repetitive trauma, and so this one episode is similar to a 180-pound person being in a car crash at 65 miles per hour. Is there damage to the nervous system? Again, we can stain these brains. This is a sleep factor that regulates sleep in flies. It's conserved in people, and I think you can see the one on the far panel is damaged. And more importantly, we can look at things like sleep. So I brought a sleep monitor here. We can, again, circadian cycle was clarified in flies, and we can go ahead and look at activity profiles, and I think you can see that we have a normal day-night sleeping and running around pattern. If you give them 5 injury bouts, they start to lose it, and 10 injury bouts, it's looking even worse. And basically through some very complicated software that we've been given by a collaborator at UCSD, we can pick it apart, and it actually boils down to they have sleep fragmentation. So I think, and by the way, we have both mutants that are fly lines that are exquisitely trauma sensitive, and we also have flies that are resistant. So I'll leave it at that, and any questions, I'm happy to answer. So, yes, go ahead.
So what is the modified diet?
Well, there's several. So the classic one has been caloric restriction, which is, nobody wants to do that. You know, just like, yeah, you just wish you would be kind of
2 days of fasting a week or something like that?
For people, it's a little bit different for flies because their metabolism is so different, but we can have it down to like 8 hours 3 times a week. I think for people, I think it's been down to 2 days a week, where you know, men can consume maybe about 500 calories, women about 400 calories. So lots of fruits and veggies, and yeah, so.
What is the condition that has to be satisfied to assume that I behave similar to this tiny fly?
Well, I don't know you very well, so that's going to be kind of hard to tell.
Let's say for human beings.
For human beings, I think the big thing is, what I think is going to come out is anything that's cardioprotective is going to be neuroprotective as well. And I think there's a wealth of information, especially with diet, that anytime you calorically restrict worms, mice, yeast, probably mice, rats, monkey studies wasn't, you know, they didn't extend lifespan, but they look great. There's been a wealth of information that you know, maintaining a really healthy diet and not developing type 2 diabetes is nothing but beneficial for you. And exercise is probably A-okay too.
If I could follow up on that, if you take some drug that we know what it does in humans, and now you apply it to flies, the question is, can you mimic
Yes, we can.
aspects of the
When
behavior
I have
that
that
you see in
data,
humans?
I'll just get it pulled together and publish it.
Could you give us an example of what kind of drug that might be?
Metformin.
Okay.
We've had really good luck with metformin. We
Which-
also have some compounds that I have with my collaborator at the Salk Institute, and those are looking really quite nice. I think Brandon's doing the aging on those right now, but we have behavior and things like that, so they all kind of seem to be doing things slightly differently. Metformin is extending lifespan, lowering neural aggregates, but behavior is kind of, so the behaviors are kind of a little bit problematic with those. and it has to do with how it functions. It's decoupling the electron transport chain in mitochondria a bit. So, go ahead.
So wasn't there just a paper that came out on the drugs that were intended to be impacting plaque formation and cognitive abilities? They were disappointed by the outcomes.
Oh, yeah.
And so
Yeah.
does your data at all address plaque formation if you're able to bump up autophagy?
By the time you're making plaques, those are extracellular deposits. And then that was mentioned in the 60-minute study, that they think that they've been attacking this whole problem way too late. And so they're working with a family that has early onset familial Alzheimer's. They can do a genetic test and know who's going to have it. And they're doing brain scans really early, well before people have symptoms, and they can see physical changes. And so they're starting to actually go back and try to do things. They were doing an immunotherapy where they're trying to clear out amyloid from people with and I'm really concerned about that. I mean, there's some really serious complications associated with that. It works great on mice, but I don't know. It's going to be, and again, the whole field has really focused in on amyloid, and that may be just something that comes up that's pretty problematic, but may not be causal for the disease. And so a lot of times when they do postmortem, when they're looking at plaque load, often it doesn't correlate with the level of dementia people see. And inflammation seems to be a huge problem. And again, diet, keeping type 2 diabetes and other kinds of metabolic syndrome problems down and low is nothing but beneficial.
Thank you.